Welcome. This EuPathDB tutorial will explain how to use the DNA motif pattern search. All EuPathDB databases are built with the same architectural design, so they look similar to each other, and features or search types are the same across all databases. This tutorial will use plasmodb.org to demonstrate the DNA motif pattern search type. The DNA motif search returns a list of IDs for genomic segments whose DNA contains a match to the motif or recurring DNA pattern that you specify. Let's go over how EuPathDB defines a genomic segment. EuPathDB uses the term genomic segment to refer to user-defined genomic regions that do not exactly correspond to universally accepted genomic objects such as genes, EST, or SNPs. Genomic segments have a known chromosomal location with a defined base pair start and end point and are not restricted in size. They can be as large as a chromosome or a region that includes several genes, or they can be as small as a 6 base pair motif. The genomic segment ID contains information about organism, chromosome, location, and strand. All DNA motif searches are initiated from the Motif form on the page Identify Genomic Segments based on DNA motif pattern. There are several ways to reach this form. One way to reach the Motif form is from the home page center panel titled Identify Other Data Types. Expand the menu item Genomic Segments, then click on DNA motif pattern. The DNA motif form can also be reached through a series of mouse-over menus. From the home page header menu, mouse over New Search, then search for genomic segments, then click on DNA motif pattern. The DNA motif form can also be reached from the All Available Searches page, which is found by clicking on the last option in the New Search mouse-over menu, or is found by clicking on New Search. Now we are ready to perform a search. The DNA motif form asks you to define a sequence that you are interested in. The search will compare the sequence to those in the databases and will return a list of segment IDs that contain your sequence. Let's search for the EcoR1 restriction enzyme site. Its sequence recognition site is GAATTC. The IUPAC nucleotide symbols are supported in motif descriptions. Complicated motifs that allow base substitutions and variable lengths must be entered in Perl-style regular expressions. For a help page on Perl-style regular expressions, follow the link below. Enter the DNA motif in the box labeled Pattern. You can choose to search for your motif in one or more organisms represented in the database. Click Get Answer when you have made your selections. This search returned 6,164 DNA segments which matched the EcoR1 DNA recognition sequence. The result page contains a graphical representation of the search strategy and a list of segment IDs that define the species and chromosomal location of the motif occurrence. This search strategy identified all EcoR1 sites wherever they are located in the genome. To find EcoR1 sites in relation to genes, the results of this motif search would need to be combined with the results of a gene search using the co-localization function in the Add Step pop-up of the search strategy. To view a tutorial on this powerful search strategy technique, click here. Thank you for watching this EuPathDB tutorial. Please contact us with any questions or suggestions.